So for the next time period in this class, I want to talk about some of the three-dimensional aspects of chemistry. Now, we saw some of this in previous courses, such as Foundations, where we talked about the shapes of molecules. But in this class, I want to get a little bit more into that and into why it matters. So where we should start is, what is the geometry of carbon? And the type of carbon we're talking about here is a carbon that has 4 single bonds, because that's what everything in this chapter or this section is going to focus on. So what is the geometry of the carbon, for example, in methane? So this is a molecule, the center atom, that has 4 electron domains. And no lone pairs. So when we're in that situation, what is the geometry of a molecule like that? This is what we refer to as tetrahedral. Now, what does tetrahedral look like? Tetrahedral, when we're in 2 dimensions, we represent as central atom. One group points up, one off to the side. Then we use this wedge to indicate that there's a group coming forward, and this dashed line to indicate a group that's pointed away from us. So what does that look like? And so if you've purchased a model kit for this class, that's where this is going to help you. So we can see our tetrahedral shape here. You can see there's one group, one atom that points up, one that's in the same plane but off to the side. We have this red one that's coming out towards you, and this blue one, which is a little bit harder to see but points away from you. So why is this something that should be of interest to us? Why is this something that matters? Well, this gets us into a topic known as chirality. And it's probably easiest to introduce chirality with an example. So here we've got pictured 2 molecules, and if you look very carefully at their structures, you'll see that they have the same formula, but not only do they have the same formula, they have the same connectivity. So the way that the atoms are connected is also the same. So what's different about them? What's different is right here. And what's different about them is that the direction that the atoms are pointed in. And that might not seem like a big difference. Does it matter if an atom is pointing up or pointing back? But before we come back and talk about this molecule again, let's look at a couple of much simpler molecules. So this is the model that I just showed you. And this you can see is a similar molecule. And what I want you to notice, I'll show you this one first. It has 4 groups on the carbon. One that's blue, white, red, and yellow. Here's my second one. Same 4 groups attached. Red, white, blue, and yellow. Are they the same molecule? So if I arrange them in this way, is there anything you notice about them? And this is where it's definitely going to help if you have your own model kit, because you could put 2 similar models together and follow along better. But are these the same molecule? Well, if they are, then I ought to be able to put one on top of the other. In other words, they make them superimposable. Now, from the way I'm holding them, you can see that they're mirror images. We could put a mirror right between them, and if we looked at the mirror image of this one, we'd see this. So what happens now when I try to superimpose them? Our blue atoms line up, our yellow atoms line up, but the red and white ones don't. All right, let's line up the red and white ones. So now we can see they line up, but the blue and yellow ones don't. All right, what if I line up the red and the blue ones? You can see they line up, but the white ones and the yellow ones don't. And we could go through every potential pair of possibilities, getting 2 of them to match up, and we would find immediately that the other 2 don't. So what that tells us is that these 2 molecules, which look very similar, it turns out are not the same. And that's the same thing happening here with thalidomide. So is there any way that you could get these 2 molecules to be the same? Well, it turns out that can only happen if you actually break some of the bonds and then form new bonds. So again, same formula, same connectivity, different 3D arrangement. Cannot be superimposed. Now, in the case of thalidomide, what did that mean? Well, thalidomide is a drug that was prescribed quite a while ago. I want to say that in the 1950s, 1960s, as a cure for morning sickness. Now, it turns out the molecule on the left works, and works very well. But if you take the compound on the right, it turns out it's a pretty severe teratogen, or causes severe birth defects. And while these are two different molecules and you have to break bonds to interchange them, it turns out in our bodies we actually have the ability to do that. And so even if you made up a drug that was only the one that's safe, it turns out it's not safe because it will form sufficient quantities of the harmful one to cause problems. And there's many, many other examples like this. So the following is also a chiral molecule. It's one that we've seen before. This is ibuprofen. And we can make another form of ibuprofen where the only thing we change is the direction that this methyl group is pointing. It's either pointing forward or it's pointing away from us. Again, we could build models of them, and we would find that we can't superimpose these two models on each other. Does it matter? Well, it does. This one on the left is what you want. This is what'll cure your headache or whatever reason you take ibuprofen for. This one on the right is pretty much useless. Won't do anything for you. And so this idea of chiral molecules is something we run into in many, many places, but especially biologically. Almost all of the molecules that you can think of biologically that are important, DNA, 19 out of the 20 amino acids, proteins, carbohydrates, steroids, they all have these three-dimensional aspects to them that are highly important. So a chiral object or a chiral molecule we define often in another, we're going to use another definition to define it. So can anyone think of something else that you might run into? Two objects that are mirror images of each other, but are not superimposable. The answer is yes. We do have that. But it seems that my camera has frozen. So where do we have something like that exists? Well, look at your hands. Are your hands mirror images of each other? If you put them up next to each other, we see that they are mirror images. But you can't superimpose one hand on top of the other. And so a chiral object, in fact, the word chiral is derived from the Greek word for hand. But a chiral object, we say, is one that possesses handedness. because it's like our hands. Or we can say two objects that are non-superimposable mirror images. As we said, almost all biomolecules are chiral. And so we've used the term isomer before, but now we're going to define a specific type of isomer, which is a stereoisomer. These are molecules that have the same connectivity, but a different 3D arrangement. So how do we identify a chiral carbon or a chiral molecule? Well, we can identify it in the following way. Any carbon with 4 different groups attached is chiral. So this carbon would be chiral because it has 4 different groups attached to it. Trying to move this over. But as soon as 2 of those groups become the same, it's no longer chiral. Now, when we say the same, that means they have to be exactly 100% the same. So are these two, is this molecule chiral? So we have a hydrogen, which is clearly different from the other 3 groups, which are all carbons. We have a 1 carbon group. We have a 5 carbon group. And we have a 7 carbon group. Those are different. And so because those are different, that means this is chiral. And when we say different, we mean any point of difference in the molecule. So we could have 2 chains that are both 100 carbons long, and if there's a bromine on that 100th carbon on one of the chains and not on the other, that makes them different. All right, so that's how we establish if a carbon is chiral. A carbon is chiral when it has 4 different groups coming off of it. What about a chiral molecule? Because those are 2 different things. When is a molecule chiral? So we're going to look at 3 different situations. 0 chiral centers, or chiral carbons. 1 chiral carbon. And then more than 1 chiral carbon. So the easiest of these 3 situations to deal with is when we have exactly one chiral carbon. If we have exactly one chiral carbon, then the molecule is chiral, the entire molecule. If we have no chiral centers, then the molecule is usually not chiral, or typically we use the terms achiral. So achiral, lacking chirality. But notice I use the word usually. So does that mean that there are chiral molecules that don't have any chiral carbons? The answer is yes. They're not terribly common, but they do exist. And what about more than one chiral carbon? So if there's more than one chiral carbon, the entire molecule is usually chiral. But there do exist, and do exist actually a fair number of molecules that have two or more chiral centers, but are not chiral. All right, so before we go any further, I want to do some history. And that's because I just think the history of chirality and how it was discovered, there's some pretty interesting stuff, and it also helps to give us a better idea of what this whole idea of chirality means. So knowledge of chirality has actually been around for a long time. In fact, the first observation of it was before 1850. And the person who was the first to observe it is someone that you might be familiar with from other places. It was Pasteur. And you probably know of Pasteur because the process of pasteurization for milk is named after him. But it turns out he was also the first one to observe stereochemistry. And how did he discover this? Well, he ran into this molecule. This compound is known as tartaric acid. And it's actually a byproduct of when you make wine. And what he was able to do was he found another sample of tartaric acid. That other sample of tartaric acid had come from a lab. And because the way these two compounds formed crystals, he was able to look at the crystals, and he realized, I think pretty amazingly, that the two types of crystals couldn't be superimposed onto each other. Now, this is not something we're ordinarily able to do. So the fact that he was working with a molecule where he could do it is pretty remarkable. And in fact, what he was able to do is even more amazing. He was actually able to look at the tartaric acid molecules under a magnifying glass, and using tweezers, he was actually able to separate the two types of crystals. So what he decided to do with them, among other things, was he dissolved each one of them into water and placed them in a polarimeter. So what is a polarimeter? So if you think about what light is consists of, it vibrates in all possible planes. But in a polarimeter, you're able to polarize the light so that it only oscillates in one plane. What Pasteur did was when he placed each of these compounds into the polarimeter, both of these compounds rotated the light, both the one he got from the winemaking as well as the one that he got from the lab. The surprising result was one of them rotated the light clockwise. That was the one from the winemaking. And so that one became known as plus tartaric acid. The other sample, which would later be determined to have this structure, rotated the light in the opposite direction, or counterclockwise, and so is known as minus tartaric acid. And so over time, scientists eventually realized that molecules that were considered to be chiral all had this property, that if you put them in a polarimeter, they would rotate the plane of the light. If you put an achiral molecule into your polarimeter, it doesn't rotate the light at all. All right, so that's where Pasteur leaves us off with this story. But back in the 1800s, it was pretty hard to figure out what was going on because things such as the electron hadn't been discovered yet. And so 1874, two other scientists came along, Lebel and Van Hoff. Now, what happened to them is something that, while certainly still can happen, definitely doesn't happen as much today as it would back then. They both looked at Pasteur's results as well as other things, and they both came up with an idea. Not together. They both independently came up with the same idea. Because if you think about it, 1874, there wasn't quite the rapid communication we have today, and quite feasible for multiple people to have come up with the same idea working completely by themselves. Van Hoff would later go on to actually win the first Nobel Prize ever in chemistry in 1901. But the idea that they proposed based on Pasteur's results was actually fairly radical. What was it? They proposed that carbon was tetrahedral. And now, that's something we know to be true. There's no dispute of carbon being tetrahedral. But not everyone bought into this at that time. And so another chemist, Hermann Kolbe, wrote the following about Van Hoff and Lebel's proposal. Not long ago, I expressed the view that the lack of general education and of thorough training in chemistry was one of the causes of the deterioration of chemical research in Germany. Will anyone to whom my worries seem exaggerated please read, if he can, a recent memoir by Herr Van Hoff on the arrangements of atoms in space, a document crammed to the hilt with the outpourings of a childish fantasy. This Dr. Van Hoff, employed by the Veterinary College at Utrecht, has, so it seems, no taste for accurate chemical research. he finds it more convenient to mount his Pegasus and to announce how, on his bold flight to Mount Parnassus, he saw the atoms arranged in space. Well, as we know now, turns out Van Hoff was right, and Kolbe, who was actually quite the successful chemist on his own, sometimes is more well known for those comments than for others. Now, Pasteur used, and for a long time, chemists used these plus minus to identify each of the two compounds, the two possible isomers. But there's a problem with doing it that way. And the problem is, when we do it that way, that relies on the way that it rotates the light in the polarimeter. That tells us, unfortunately, absolutely nothing about the structure of the molecule. And so we needed a new bit of nomenclature, a new system to name chiral molecules that would tell us about the structure and not just about the way it rotated light.